Okay guys, welcome back. We are going to put a cap on the subcellular structures. We've been looking at organelles and we kind of flew through a bunch of them last time. We talked about the nucleus, we talked about lysosomes, we talked about rough ER and smooth ER. And so I wanted to revisit it really briefly here because I think I went through quite a few last time. And just to kind of give you an idea of how I would study for this and simplify it a bit, right? When you're doing your objectives, you want to have something simple so you can recall these kinds of things during the exams. So I'm looking here at an image on the left that you would get in chapter 2 of your book. And they're beautiful images. We really have some great resources when it comes to your publisher here. And then this one down a little lower in the screen shot here is from chapter 4. Now we aren't really doing chapter 4 stuff quite yet. That will come in the second module. But you don't have to be an artist to be able to draw something to allow you to have a visual that would help you to trigger some of these memories when you're going into your quizzes and your exams. A lot of times drawing something, as rudimentary as it may be, will help you to remember just the act of doing it, the physical act of doing something like that, rather than just reading words on a page, okay? So what I did, and you're laughing probably now, but this is what I put together this afternoon. I did it with my mouse because I don't have any fancy tablets or anything to draw with, but you can do this on a piece of paper, right? What I would want to do is I would want to get as much into a picture as I can to answer as many objectives as I can. So what I've done here is I've drawn basically an image of a cell here, okay? This entire thing is a cell. And we've got our plasma membrane here. And what I've even done here is I've chosen one section of this plasma membrane to focus in on, and we're looking at our phospholipids, and we're looking at transmembrane proteins. They may do different things. They may actually be a channel allowing for passive transport, right? Facilitated diffusion, for example. It could be one of these sodium-potassium pumps, and it does not have to be pretty, right? I mean, this is not stuff that is really that great artistically. And what I would do, I didn't finish this, but what you could do is you could continue as much as you need to put into this plasma membrane all the things you need to remember about plasma membrane, stuff we've gone over. You could put endocytosis in there, put a little image in there. You could put in all the functions. You could even put some of the intercellular junctions, right? Those cell adhesion molecules like tight junctions and desmosomes. We saw those on the previous slide here. I don't know if you noticed, but desmosomes were here. Remember, the desmosomes are for strength, tying two cells together. They've got these roots, right? And you can see those yellow roots that are actually proteins, fibrous proteins that hold those cells together. Well, you can do that here as well, okay? But what I wanted to focus on with you guys here and now are the organelles. Incidentally, here we have some microtubules I fit in there. But on the inside we have the organelles, right? So you can put in your nucleus, right? You can put in a rough endoplasmic reticulum. So down here we have a membrane, kind of a labyrinth of membrane going back and forth. And then I've got in red, each one of these would be a ribosome, right? That's what makes it rough, is that these ribosomes are kind of etched, kind of studded all along that membrane. And you could write rough endoplasmic reticulum. You can write the function right in your image, or put a number there, right? You can have 1 for nucleus, 2 for rough ER, 3 for the Golgi apparatus, right? All of this is meant to be the Golgi apparatus. And then on a side note, have the functions, the name and the functions of 1, 2, 3, 4, and so on. Here you can see the Golgi apparatus and how it's receiving various proteins, let's say, that were made at the rough ER, right? Because proteins are made by ribosomes. And so these ribosomes would have made proteins in this rough ER, and then they travel in these little bubbles to the Golgi Apparatus, which is our FedEx, right? Or UPS. It's going to tag and sort these proteins for distribution, transportation to their appropriate locations, right? Some of them might be enzymes, and they go to the lysosome, the recycle center, right? Some of these might be for secretion, for transport to the exterior of the cell. And so you see that happening down here. I have a little bubble that's going down, and then it's performing exocytosis, dumping a bunch of these things outside of the cell. Okay? So you can get as many things as you can on this one image, and then off to the side or on a separate piece of paper have your functions. Here's the smooth endoplasmic reticulum, and we say that one would be smooth because it doesn't have the roughness from the ribosomes attached. No ribosomes means no protein production there, but we talked about smooth ER a little bit last time with its lipids and steroid hormone production and things like that. Something we didn't talk much about were some inclusions. Inclusions are non-membranous structures in the cell that do exist. One example of an inclusion inside of a liver cell would be glycogen. Glycogen is just a big polymer of glucose, so it's a polysaccharide, and it's a complex carbohydrate, we would say, right? So inclusions might include something like glycogen. They're not actually contained within a membrane like a lot of these other organelles we keep talking about. Well, we have one more to talk about, and it's an important one, and that happens to be this weird one here with what seems to be two membranes, one kind of circuitous membrane on the inside and a membrane on the outside. This is the mitochondria, and that's what I'd like to talk about with you a bit, and then really dive in and see what is the importance of the mitochondrion in our last clip, right? All right, so the mitochondria, probably a lot of you remember from maybe even grade school, but at the least maybe in biology in high school or 8th grade, they talk about mitochondria as being the mighty mitochondria, right? Just a cute little way to remember it. It's like the powerhouse of the cell. This is, in other words, giving the cell a lot of energy. Now, so we think about this as kind of the power plants of the cell, and we've been talking a lot up to this point about this molecule called ATP. And I haven't really wanted to go into it in great detail other than to tell you guys that ATP gives energy, okay? The ATP gives the energy for pumps to work in the membrane, for example. The ATP is necessary for endocytosis and exocytosis, these active processes for getting things across the membrane. So ATP has come up a bunch of times, but now we're going to have to dive in a little deeper to see how it is that ATP and the supply of that ATP has something to do with this very important organelle called the mitochondria. All right, so when we look at the amount of any organelle inside of a cell, it starts to explain to us how that particular cell functions. If we see a lot of smooth ER in a cell, like we see in liver cells, well, there's probably some lipid processing or detoxification role being played by that particular set of cells in that organ. Or if we see a lot of sodium-potassium pumps, well, that's probably going to produce quite a large membrane potential, right? A difference in charge across the membrane because of the way that that pump works. Well, with the case of mitochondria, if we see a lot of mitochondria in a cell, we know that that is an energy-hungry cell. It's a very busy cell, and it needs a lot more energy than one that doesn't have as many. But we also see some pretty strange and unusual things when we look at the mitochondrion. We see that there are, in fact, two membranes. Okay, so here we have one inside membrane, and then we have this outer membrane. And this we don't see very many other places except for the nucleus, this double membrane. Lysosomes, smooth ER, a lot of these others, only one membrane. And even the plasma membrane is a single bilipid layer. Okay? When we look inside of the mitochondria, this is even more interesting, we find actual DNA that is mitochondrial DNA. So the mitochondrion has its own DNA separate from the nuclear genetic material that we call the control center of the cell. So this kind of has some of its own control built in, and ribosomes are even found inside of this organelle for protein production. So we've got the code for proteins with the DNA. We've got something to make proteins in here, okay? And if we zoom in on some of these inside membranes that I started talking about here, we find that there are enzymes, proteins in other words, that are attached to these membranes, that many of which are coded by the DNA within this cell. So it's some very unusual things. And when we take a step back and start to examine the DNA inside of these subcellular organelles and compare them to DNA that's out there in other living organisms like bacteria, we see some striking similarities. And so this has led the scientific community to understand and believe that the mitochondrion is actually kind of a holdover of a cell that most likely lived on its own in the past. Okay? So a large cell, namely this entire animal cell that we might have in our body, actually contains what used to be much smaller cells that were free living, that didn't need to be inside of another cell. But now we have this symbiotic relationship, okay? This little cell here that used to be out on its own, once it was taken in and housed by a larger cell, it was protected, okay? And in turn, what we find is that these mitochondria give a lot of energy to the cell that contains it, okay? So energy is a big deal when it comes to the mitochondrion, and we're going to use the entire last clip to talk about a process called cellular respiration that the mitochondria are very much a part of. And that will be the last bit that will address, I think it's objectives 29 and 30 definitely 30 that I skipped earlier, okay? So we'll leave it at that, and we'll catch up on our last clip very shortly.